Ya. Adelante, Ricardo.
Buenos días. Bienvenidos a la sexta sesión del Foro Virtual Internacional de Tuberculosis 2021. Les recordamos a todos los panelistas y asistentes que esta sesión está siendo grabada y transmitida por Zoom y simultáneamente por Facebook, YouTube y el canal Imagen TV UABC. Así también se cuenta con el servicio de interpretación inglés-español y damos la palabra a Carlos León para que dé las indicaciones para seleccionar el idioma inglés. Tres. Bueno, para mayor información pueden consultar la página de Remit TV.
And you can go to the RemitTB website, www.remittb.org. The SEPA unit at UABC is helping us with this transmission, and we thank them for their excellent work. This forum is being brought to you thanks to different institutions, our internalization program at our School of Health in at UABC, the U.S.-Mexico Border Health Commission, the TB Interdisciplinary Research Center, RemitTB, the National Center for Control of Diseases, NFRS, the Ministry of Health in Mexico, and the National Network of Research to Prevent and Control TB in Mexico. It is important to note that the objective of this forum is to provide you access to biomedical collaboration and research internationally for the prevention and treatment of TB under COVID-19 and be prepared for potential new pandemics. We want to recommend to those of you who follow us, or also want to let you know, rather, that you will be getting a QR code for each of the sessions, and if you attend 6 sessions, at least you will get a certificate of participation. And we have excellent speakers today. We have Rafael, Laniado, Laborín, as well as Jordi Torrelles. Each talk will last 45 minutes, and we'll have 5 to 10 minutes for Q&A. We will start our session with Dr. Laniado, who is in charge of the Lab in Tijuana and the Green Light Committee of the Americas, as well as the Pan American Health Organization. His talk is entitled Practical Guide for Interpreting Molecular TB Resistance. Thank you, Ricardo, and I want to thank RemitTB for the invitation. I have no conflict of interest to divulge regarding the equipment that I'm going to talk about. I will start by saying that 1 in 5 patients of the nearly 500 million patients eligible for TB or MDR TB treatment receives the right regimen. These errors in treatment are associated, of course, with higher rates of failure and death. MDR TB approaches need to include a large enough number of effective medications. And it's a significant challenge for doctors around the world, and Mexico is not the exception, because many times we have to make therapeutic decisions with very little information when it comes to drug sensitivity tests. The WHO has recommended quick tests to identify drug-resistant TB patients who could benefit by the new treatment alternatives, which fortunately we finally do have available. Xenotypic tests, which is what we had traditionally used, are not appropriate to that end because TB grows very slowly in petri dishes, and genotypical assays that provide results within hours, but day at most, are accurate, are becoming more automated, which is a huge advantage, automatization, because it allows us to have them at the same site where we're seeing the patients. We don't need to send them out to a lab necessarily. And they are also affordable today. They are a viable and effective avenue for therapeutic decision-making. Today, TB diagnosis needs to be done through genotyping or genotyping. One of the issues that we face as doctors, however, are the differences between phenotypical and genotypical methods, and we don't know who to believe. It has been discovered by a study that was recently published in Lancet that reference lab results and local lab results may have as much as a 20% difference. In a logistic regression model, the probability of death was 7.3 times higher for patients with differences in their results. That could lead to an inappropriate treatment. So the question that we ask ourselves as clinicians is whether we could come up with some sort of recipe for interpreting results. That is, for the lab to send us a recipe so that we don't have to try to understand what's going on. And I will also show how and if we can have that recipe or not. Part of my presentation focuses on sensitivity and specificity of tests, false negatives and false positives, heteroresistance, very briefly, mutations detected and inferred, and finally, disputed ones. So the question that we have to ask ourselves to begin with the conversation is how accurate our everyday tests are. And to do so, we must talk about the specificity and sensitivity of these tests. Let's start with GeneXpert. GeneXpert has been recommended now for a decade by WHO for the quick diagnosis of rifampicin resistance and TB. When we compare it to conventional methods, it is of course better. Now, when we assess bacillus, you would require between 5 and 10,000 bacillus per sample. And GeneXpert is accurate, but Expert Ultra is even better. Expert Ultra has a sensitivity that is very similar to cultures, which is the gold standard. So while it is very sensitive, the issue that we have, as you can see in this graph, is the time to results. It takes much longer. While the Expert and Expert Ultra techniques gives you results within hours. The G4 can have 116 culture causing or UFCs per millimeter, but the Ultra has a detection limit of plus or minus 15. So what happens when we talk about sensitivity and specificity of Expert versus BK? Well, it depends on how we use it. If we use it instead of bacilloscopy, as the WHO indicates, to substitute it, then it has pretty good sensitivity, 88%, and very high specificity. So it's very good. Bacilloscopy has a sensitivity of 50 to 70% at most. And people say, well, you thought that it was so good to do. Sure, but 70%, it's not great. But if you do a bacilloscopy on 10 patients who have lung TB, we're going to tell them at the health unit that, they don't have TB. But if we do it with Expert, in 7 out of 10, we will know that they have it. Now, when you have a positive bacilloscopy, we have the same specificity. Now, in patients with HIV, the sensitivity is close to 80%, since most of them are, and to detection to rifampicin, it has a very good sensitivity of 95%. That is to say, out of 100 TB patients that are resistant to rifampicin, Gene Expert would detect 95 out of the 100. The other test recommended by the WHO, the other quick genotypical or genotyping test, is the molecular line probe assays, or LIPAs because of their initials in English. It's the one that is most used in our region is genotype. It is a test based on microbacterial strips with and without mutations. So what is the sensitivity on version 2 of genotype MTBDR plus? And you have direct and indirect. Indirect is from the culture, and then direct is from the patient themselves. Now, as far as resistance to acinicide, it's around 98, 99%. Now, for rifampicin, we sample a gene, and when it comes to isoniazid, there are several different genes, and that's why it has a slightly lower sensitivity. Obviously, the rate of undetermined results is higher in patients with negative vacilloscopy. Now, when it comes to quinolone resistance, which is very important to us today, the sensitivity is around 86% for the version 1 genotype. And for injectables, which we try not to use, which we leave for patients that have no other alternatives, it would be 87%. Now, for what used to be known as XDR, so resistant to injectable quinolones, now the definition has changed, obviously, genotype is not great. But we're not too concerned with that because, again, we would rarely use those antibiotics today. So how does Gene Expert work? Expert is a real-time PCR test that creates copies of specific segments of the DNA in each cycle. So it doubles with every cycle. The probes, it has 5 probes, are joined amplicones and create signals. And the probes will determine whether specific sequences are present. Now, each of the experts has 5 probes. And each is complementary to a different sequence. And here we see 5 probes and the SPC, which is one of the ways that experts determines whether there's inhibition in the PCR process to invalidate the test if need be. These 5 probes encompass the central region, the full region of 81 pairs for the RPro-B gene. And it has 5 base pairs, and what the G4 cartridge tests for is this central area, which is hyperrecombinant of 81 base pairs. These probes are superimposed to cover this whole central region. So what happens when the reaction starts? If in the sample there is a sequence with a mutation, as we represent here on the image, so the probe is sort of bent, and then we have the fluorescent material, and it doesn't hybridize. So the arms remain together. So the fluorophore is not released, and then there's no signal. And then we have a wild type sequence without mutations, and it becomes hybridized. The fluorophore is released, and then it issues a signal. And this is the report that you get from the machine. If you run an expert test, this is the report that will come out, which has the patient name, the type of sample, the result, whether it was detected or not, Mycobacterium TB, and whether it's resistant to rifampicin or not. Interestingly, it also gives us each of the probe results. Unfortunately, some labs in Mexico have opted to transfer the results to their own lab format. I don't know if they want the stamp of their particular lab or why it is, but the risk, although it's a small risk, is an error in transcription, which has happened. We would rather get this original report. And how does it work? There's a detection threshold for the cycles in the PCR reaction test, which is called the cycle threshold, where the signal crosses that detection threshold. So, and depending on how many cycles it takes to be detected, we can determine the DNA load in the sample. So, after 16 cycles or less, we say that it is a high load. If it's between 17 and 22 cycles, we say that it's a medium load. Between 23 and 28, a low load. And if it takes over 28 cycles, then it's a very low load. So what does it mean if the probe doesn't hybridize? There are three possibilities. One, there could be mutations. The mutations may or may not be clinically significant, as we will see, but there are mutations. Now, it could also be that there wasn't sufficient DNA or there's no DNA. For example, in the negative smears, you might not generate sufficient copies of the DNA, but we may have a positive smear, but a negative expert. This strongly suggests that it is a non-TB Mycobacterium, since expert is so good at detecting. And finally, there could be an inhibiting substance that inhibits the amplification. Expert has an internal control to detect that. The SPC signals indicate if there is or isn't an inhibiting substance. And if there is, it invalidates the test. Now, these curves are usually not sent to us by the labs. As you can see, none of the probes hybridized, so no MTB was detected. None of the probes issued a signal. The STC has a signal. If it didn't, the test would be invalid. So, in this patient, we did not detect TB. And here we see the 5 probes, and then we see the SPC showing that this is a valid test. All 5 probes issued a signal, they're hybridized. Now, as far as rifampicin sensitive, there was no mutation because the difference between maximum and minimum is less than 4. Here you see probe D has 33.9 and probe A, which is the lowest, has 31.8. And SPC has signal, again, indicating that the test was valid. Finally, in this case, we see that 4 of the 5 probes hybridized. This one did not, but the SPC did. So we see the difference between the highest and the lowest is much higher than 4. Probe D is at 24.8 and probe E is at 0. That the most common mutation detected is this one here, and it is known because there is a list of mutations associated to each of the probes. Now, expert then can detect mutations. The most common, as I mentioned, is through probe E, and the second most common are detected by probe D. And something that happens often in clinic is detection in the B probe. 1 in 5 could be a silent mutation, one that doesn't have a clinical significance. Something important, because we've seen this when we visit other countries, is that they use the expert not just for diagnosis, but also for follow-up. And in my opinion, that's a mistake. The assay is incredibly sensitive, and it can continue to identify DNA fragments even if the bacillus is already dead. Here, as you can see, after treatment started, the bacilloscopy, it starts reducing, and then the Lowenstein-Jensen medium. But 3 out of 4 patients who have negative cultures continue to test positive in the expert because it detects DNA fragments. So the assay should be used exclusively for diagnosis, but not for treatment. Sometimes in follow-up, we do it just to make sure that they didn't develop rifampicin resistance during treatment, but that would be the exception. Now, the predictive value of the perfect tests that we use, because they're all imperfect, depend on the probability of resistance prior to the test. For example, if in the region where we work, let's imagine that we're in Sweden and there is no TB resistance, the negative predictive value for rifampin and other drugs would be very high. As resistance higher, then the negative predictive value would start going down. For example, sometimes I have cases with low likelihood of being resistant to rifampicin or rifampin, but we get a result that says that it is resistance. Let's say that the patient is in Finland, where there's no resistance, and it shows a rifampin or rifampicin resistance. What could be going on? Well, it could be a new case. It happens, where they have a low resistance to rifampin. It could also be an ID or report error. Again, that's why we need to get the actual result from the machine instead of transferring the information to their lab format. Now, from the clinical perspective, it is unlikely, or epidemiologically speaking, that the person would be resistant to rifampin, but expert is telling me that it could be. Again, the test is good, but it's not perfect. So you can do a second sample and then adopt the second result as the correct one. The likelihood for a false positive test on a genotypical test is very low. It's between 0 and 2%. Now, how about genotype? How does that work? Again, it is a microcellulose strip with DNA fragments. So with what wild type genes with that mutations, and for specific mutations that were chosen based on the frequency that we see them with. In this strip, for the second line, we have wild type fragments for GRA, for quinolones, and we also have mutation fragments for quinolone. So how are they detected with online strips? Well, they can be detected in two ways. If we use amplicons or MUNT strips, we say that they are detected probes. However, if there is no hybridization, that is, if the amplicons do not with the wild type probes, then the reaction after hybridization leads to the development of colored bands in the microcellulose strip. In this case, for example, we see a sample from a patient, and we have the genotype strip, and here see how all this the white type for quinolones reacted, both for GRA and GRB, as well as the RRS and ES. However, none of the mutation ones reacted. So this genotype would be reported as no resistance mutations are detected or inferred in none of the genomic regions included in the LPA. So there were no mutation bands that are. Now, what happens with silent mutations? Those are the ones that are not significant clinically, but that do exist and can be detected. In this case, the codon is modified, but not the amino acid. And therefore, the protein that codifies it. For example, with rifampin and silent mutations, the probe doesn't hybridize, indicate a mutation in the nucleotide, and then say, oh, the expert made a mistake. No, the fact that it is a silent mutation, is a different thing. Here we see false positive rifampicin resistance on expert MTBRF caused by a silent mutation on the RPOB gene. And here we see that the analyzed strain has TTG and this one has CTG, but the amino acid was not affected, and therefore the protein that codifies it. So how frequently do we see it? Well, depending on which study you read, that could be 2.5, it could be 4%. This, which was the largest study, it was less than 0.8%. So what happens with false negative? Well, we were saying that GeneXpert gives us 95% sensitivity. That means that 5% of the mutations will not be detected because they are beyond the hypercombinant region. 5% for expert and 4% in LPAs. Why? In the case of expert, because we know that the RPOB gene is much larger, and there are areas that expert does not sample. So 5% of these infrequent mutations are beyond the area being sampled by GeneXpert. And although LPAs can detect mutations that are most frequently reported, like the case of expert, there will be mutations that will not be detected because they're beyond the regions that are covered by the test. And so, from the clinical perspective, we also need to do genotypical tests. Once we have clinical access to sequencing, perhaps we won't have to do them anymore. But for now, we should still continue to do it. What are the consequences of a false negative for rifampicin or any other medication, but rifampicin in particular because it's our best medication? Well, it can increase morbidity and mortality in patients. It can lead to death. And very importantly, when it comes to public health, we could have community transmission of strain that is resistant to rifampicin. So if the patient gets his primary treatment, the treatment will fail in that whole time. They will be transmitting that resistant strain. So let's talk about heteroresistance. That is the existence of a subpopulation of microorganisms that are resistant within us, microcommunity that is sensitive to a particular medication. Now, 57% of strains are multi-drug resistant, present heteroresistant with one or more mutations that are recognized as associated to resistance. The sequencing, this is a very common. Heteroresistance to rifampicin could come out of a clonal subpopulation. So there's a subpopulation clone that is resistant to rifampin or rifampicin. And it could also be from different strains, one that is resistant and one that is sensitive. This is also called an unfixed resistance. And it precedes the total resistance where all of the bacillus will be resistant. And that is because we didn't provide the correct treatment. And so the sensitive bacillus, but the resistant bacillus continues. Now, expert is not as good for that. It is better through LPA strips, but of course, culture is better. Now, LPAs are less efficient than genotypical tests to detect resistant strains in samples with heteroresistance. So when can it be done? When it can be detected by the MOOCs. Now, detected mutations versus inferred mutations. I spoke about it briefly. The term detected resistance is used anytime that one or more mute probes identified specific mutations that confer resistance. However, inferred resistance is used when one or more or the wild type probes in gene regions that are associated with resistance do not hybridize. So in the case of inferred resistance, it is not possible to identify which specific mutation exists, but rather only the region that the mutation exists in. Let's see this example. Here, the wild type probe did not hybridize. You can see it didn't hybridize here. And the RPOB mutation did. So we have rifampicin resistance that is both inferred and detected. In the same sample, we see that the CAD-G probe did not hybridize. So we have inferred isonucleotide resistance, and this patient is then defined as an MDR-TB case. We see that the wild type probe for INH8 did not hybridize, but the mutation for INH8 did. So we see a detected resistance to isoniazide. So one is inferred. The wild type probe did not hybridize, but the mutation one did. So the wild type did not hybridize and then detected because the mutation did hybridize. That's how we get it from the National Reference Lab as to whether we have an inferred or detected resistance to a particular medication. So if resistance inferred through the lack of union between amplicons and wild type probes, it is suggested to do a gene sequence for RPOB to determine whether the patient is resistant to rifampicin. Finally, disputed mutations. This became an issue about a year and a half ago, and we've resolved it in most cases now. We have a publication from the FoVI-TB group that has been accepted. Certain mutations in the RPOB gene, called disputed, are detected genotypically. It's detected by gene expert, and it shows a result of rifampicin resistant. But the MIGIT, since it used a liquid medium, reported sensitivity to rifampicin. So then the clinician would ask, well, who do I listen to? MIGIT says that there is no rifampicin resistance, and expert says there is. So what's happening? First, we need to learn that phenotypical tests, which are the gold standard still, may underestimate the performance of genotypical tests, where false positives are not really false positives. These mutations that the bacillus, when it comes to rifampicin, affects what in English we call the fitness, how well the bacillus can multiply, which makes it grow slowly in the presence of the medication. And since the MIGIT has a software that throws out the sample after certain days, it will report it as sensitive because it doesn't reach that threshold where there's been enough duplication to show resistance. Since the minimum inhibiting concentrations for this mutated strain would be below the conventional critical concentration, and thus it reports them as sensitive to rifampicin. So the WHO this year has suggested that if we're going to run MIGIT for rifampicin to modify the final concentration of rifampicin, which is recommended 1.0 micrograms, and now almost all this year we've been running at a 0.5 to take into account those mutated strains. So what is the road before us? Well, the first question is whether we should continue investing in phenotypical methods to diagnose drug-resistant TB. I would love to say no, but we still need to say yes for now, and I will explain why. First and foremost, genotypical methods, in order to work, have to kill the bacillus to extract the DNA. And we in clinic, when we're doing the follow-up of a case, what we want to know is whether those bacilli that appear in the bacilloscopy are alive or not. And today, the only way to know it is by cultures. To warm them up, feed them, and let them grow in a medium, which will let us know whether they're viable or not. So we will have to continue using phenotypical methods to follow up on our patient. The initial diagnosis should be done through genotypical method, but there are exceptions because we still do not have clinically accessible genotypical tests everywhere. Well, you might say, well, there's sequencing. Yeah, but sequencing isn't accessible everywhere, at least not in Mexico. So we don't have genotypical tests for certain medications. For example, numesulide, clopacin. So for those, we need to use phenotypical methods, the one that we've had for over 100 years. It takes a longer time if you want, but that's what we have for now. I have no doubt that before the end of this decade, as Kennedy said, we will have genotypical tests that will allow us at point of care, and that's the beauty of the Gene Expert, that it is at the clinic where the patient comes, it's a point of care. And anyone that's halfway trained can process and use it. So I have no doubt that we will have these kind of tests at point of care for all of the medications currently in use at some point. But at least for now, we will continue to invest, not as much as we did before, but still for phenotypical methods, for diagnosis, and for resistance to medications for which we do not have genotypical tests available. Now, full genome sequencing. This approach allows us to reveal the DNA sequence, the full sequence of the tuberculosis full genome. It's about 4 million base pairs, and it adapts very well to the focus of the sequencing. The WHO recommends it, in fact, that we need to start doing sequence. Now, the assay that used to be very expensive and took a long time is quick, is reliable, and more affordable. And it includes all of the guidelines for controlling TB. It's the best tool because it allows us to diagnose. It will tell us every mutation that exists. We might not even know what some of them are for at first, but we'll learn. Also for treatment, to choose the right medication. For surveillance, epidemiological surveillance, and to do outbreak investigation when we have TB outbreaks. Now, the REMIT TB group has started doing some research when it comes to sequencing. This article in the International Journal of Infectious Diseases says the whole genome sequencing for Mycobacterium bovis to obtain molecular fingerprints in human and cattle isolates. Now, our national group, little by little, we're starting to see sequencing results being done by the Genetics Institute for our patients in clinical application. So if we have any questions as to whether our medication would or would not work. So the sequencing and its different modalities will become a tool that we'll start using more and more frequently in clinic. For those of you who are interested on the topic, there are two great documents. There's one by GLI, which is a lab diagnosis group at WHO, or the International Association guideline, which I also recommend. So, in closing, can we create a recipe? Which was my original question. And the answer is no. And even though labs can facilitate it, we can't really have just a single recipe. So in conclusion, genotypical assays are accurate. They are becoming more and more automated and more affordable, but again, they're not perfect. The easiest example was the expert, 95% sensitivity to rifampicin resistance. So if we only use that method, we will miss 5%. Now, when it comes to the TB doctor, well, we're going to have to continue studying to become more familiar with new technologies. For practical purposes today, if there is a positive result as far as resistance to any medication, we should consider that strain as resistant until such a time as we have complementary data. So you can't do a single test. So when we ask, are you doing sensitivity tests? Oh yeah, we use expert, but that's only one screening tool. You need more to have complementary information. And of course, and this is where we can have disagreements with the lab, the previous treatment history is important in decision making. If we have a patient who has received the same medication 3-4 times and who has positive vasculoscopy, then it's not likely that the medication will continue to work. And then the lab says, well, it's up to you. But here it says that the patient is sensitive to that medication. And that's when we have to look for alternative methods to have a more definitive diagnosis of the patient. So I close with that, and I thank you very much for your time and attention. Thank you so much, Doctor. A very interesting presentation. Very informative. We do have some questions from the public. And our first question says, when it comes to that 20% difference between phenotypical and molecular tests, is that due to their different sensitivities, or that one is just focused on the RPOB region? Well, the answer has different elements. First, if we're going to talk about rifampicin or rifampin, as I mentioned, it only samples a segment of that RPOB gene. So there could be differences, because if it is part of that 5% that expert does not test for, then expert would report it as sensitive to rifampicin. But if we do a culture, we could see that it multiplies in the presence of rifampin or rifampicin. So that's one of the differences. But it could also be the presence of silent or disputed mutations, and it depends on how the lab is processing the rifampicin, which should be at 0.5 instead of 1 microgram. And the sequencing, in fact, in the group of patients that I was sharing about earlier, Dr. Muñiz was kind enough to run the sequencing in order to determine if any of the mutations we found in the cases were clinically significant mutations or whether were silent mutations. And she found several that were silent. And that's an issue for the patient, because if we do not have the sequencing support, we're going to treat it as if it was treated, as if it was resistant. It will require longer treatment. Now we have Jordi, who wanted to ask out loud. Yes, I did. Doctor, I have a question for you. I've discussed this with several colleagues in South Africa and in the Democratic Republic of Congo, and they've concluded there that one of the worst things that has happened in diagnosing TB, it has been the implementation of GeneXpert. And the reason they give me, and that's why I want your opinion, is that they're finding that 63% of rifampicin-resistant cases are not being detected by GeneXpert, and they say that it is due to the fact that it is not linked to an RPOB mutation. They're also finding that people in charge of GeneXpert, and as you've said, is very easy to use. So now they're running out of inputs, and most of the hospitals where GeneXpert could work no longer works because they have no power, they have no water, they can't do the calibration and so on. And so they're finding that people cannot do vesselloscopy anymore, cannot do smears, because they've depended on GeneXpert for so long that now they don't have the people to do it. I don't know if this is something that is exclusive to Africa or whether it's an issue in other places you know about. Well, thank you so much for the question. Now, there are definite obvious reasons for what's going on in Africa. These strains that were being found as rare mutations that were part of that 5% that GeneXpert does not detect, when there's a lot of community transmission, become dominant. And in Africa, we have more than 200 per 100,000 resident rates. In the US, it's 2.8 per 100,000. Just to illustrate the seriousness of the TB epidemic in some regions of the world. And so that strain will be transmitted in the community, especially because it's not being diagnosed. They run the GeneXpert, they use primary treatment because it shows that it would be sensitive, the patient fails, and that whole time, they are transmitting a strain that will not be detected by GeneXpert. So I understand that issue. And in addition to that, technology has replaced lab technicians. So they stop using a method and then they forget how to do it. In Mexico, however, I think we have a good combination in the sense that we're still doing bacilloscopy at our lab, and all of the samples are subjected to bacilloscopy in addition to gene expert and culture. So it is necessary to retain that technical capacity, because when it comes to follow-up, it is important. Imagine that you have a patient that is that has drug-resistant TB, and he's in isolation, or even if he's sensitive, and within a month he gets negative bacilloscopies, but not so in culture. And the patient may be able to relax some of the security measures. If we're going to wait until the culture shows, the patient might be in isolation 2-3 months. So bacilloscopy helps because it's a quick screening tool and it's cheaper. It shouldn't be the definitive diagnostic tool, but it can be a quick screening tool. Thank you. Thank you so much. Okay, continuing on with our questions. Do you have an estimation as to the frequency of mutations in the Mexican population? No, not yet, but I think we're going to start with that, because little by little, we're detecting that issue here in Mexico, although it has existed in literature for 8-9 years in some articles or briefers. So we're becoming aware of it, and we need to have a national causation study. Now, groups like FoVI-TB does group research, or it could be GARAPAR, which is the resistance group, research group nationwide, so that we can get data from different parts of Mexico and then estimate frequency, but we don't know specifically today. Okay, the next question is, how many labs in Mexico have the lipase strip so that it can be used more routinely? And how far are we from doing molecular tests routinely in Mexico? Well, in theory, all state labs in Mexico, so every state has an expert device with 1, 2, or 4 modules. The issue is that some aren't calibrated, some of the modules are damaged, sometimes they don't have cartridges, or sometimes we don't have cartridges. So the gene expert's there, but sometimes we can't use it. Genotype is only being done in Mexico City. There's only 2 places, at the National Reference Lab, which full title is the National Epidemiological Diagnosis and Reference Institute. Now, being that I'm in Tijuana, I'm in 2,000 kilometers from Mexico, and even though this national lab can help us, it takes time to send out the samples, get the results back, and then sometimes the cultures get contaminated when we send them. So how do we resolve that? We need to regionalize it. And there are 2 possibilities in this regard. One, to have molecular labs in every state. Every state lab is supposed to have a molecular biology lab, but it's not doing genotype tests for TB or something simpler. Fortunately, now there's a gene expert called the 10-color expert. That expert allows you to use all the cartridges, the G4 Ultra, the new cartridges. And when they launched it, they called it XDR, but they're pre-XDR. But these new cartridges allow you to diagnose for injectable quinolone, isoniazid. And so if we have that kind of equipment, then regionally we could do diagnosis for isoniazid, rifampicin, and then injectable quinolones, which we rarely use. But we could do an MDR test within a day. So the states with higher burden, like Baja California, Tijuana is the city with the highest rate of TB in all of Mexico, and Baja California has the highest TB rate, and TB-related death needs to have that equipment. Nuevo León, Guerrero, all the states that have higher burden, and only send samples to our national reference lab for specific rare resistances. Now, can expert also be used to detect extrapulmonary TB? Yeah, it can be used. Of course, lung samples are the first ones that were used, but particularly in pediatrics, and we ask pediatricians who sometimes say, no, it's very difficult to do an etiological diagnosis in children. You can do nasopharyngeal or gastric liquid test, or then you can also do it in urine and feces. Of course, it's not, it can be 50% less concentration than in sputum. And before culture and bacilloscopy, if you suspect, it can be used with children in pediatrics in many of the cases, using spinal fluid. Spinal fluid, you know, is very accurate and expert. Unfortunately, yes, we've only heard about one case. We got a fact. It was a case that was sequenced, and they found that it was resistant to clofazimine, since unfortunately they share the same mutation gene. So if one strain is resistant to bedaquiline, it is automatically resistant to clofazimine. And that is why it is important for the National Reference Lab to be able to do these phenotypical tests for these medications and namisolid, because the new treatment regimens are using these medications. And if we didn't have these tests, then the treatment would be empirical, betting on that they would work because they're new, and sometimes it might not be. So we're doing some testing. So fortunately, at least at one location in Mexico, we do have the technology that allows us for more complex cases to have fully proven treatment. Can you talk about RNA Mycobacterium the TB test? I don't know what that question would be about. Maybe somebody from a lab could answer that question. I think that those were all of the questions that we had. Thank you so much, Doctor. Well, thank you. And now we want to welcome Dr. Jordi B. Torrelles, who is from the Texas Biomedical Research Institute, who will go ahead, Doctor. Can you see white screen now? Yes. Okay, I would like to start by thanking Dr. Centeno, Dr. Muñiz-Salazar for this opportunity to present my studies to all of you. And I also apologize because I'm going to be switching to English because I've been living in the States for 25 years. So my Spanish is very rusty.
So today I'm going to be speaking about the influence of the human lung mucosa in Mycobacterium tuberculosis pathogenesis. And one of the questions that we have is why tuberculosis is still a problem. And the WHO or the World Health Organization says that is mainly for three reasons. Why is because of the ignorance that is still out there. And as an example, in 2019, it was a survey implying 3 million people around the globe, and they asked the question, what is tuberculosis? And the main answer is a disease transmitted by the rats. The second most frequent answer was that is transmitted by smoke. Another one doesn't exist anymore because it's a medieval disease. So as you can imagine, ignorance is a big problem. Another one is stigma, and it's going to be difficult to battle this problem because, as an example, in 2018, the WHO have a press release in which said that Swaziland, that is close to South Africa, had the higher rate of tuberculosis in the world. However, some reporter that was probably sleeping in that conference release heard Switzerland. So he reported through the European Express that Switzerland was one of the major countries with tuberculosis high rates in the world. And that caused demonstrations on the streets of Switzerland. Around 12 immigrants were killed because they thought that the immigration was the cause of bringing TB to Switzerland. So as you can see, this is a big issue too. And lastly, they also say that it's due to the bad implementation of the DOD, the direct of therapy, because if it's not implemented very well, it causes the development of drug-resistant TB. And right now we are starting to see high increased numbers in the continent of Africa of XDR and XXDR TB.
Recording in progress.
And as you can imagine, seeing a recent born being with XDR TB and being injected every single day with injectable drugs is very hard to see and to treat. And just some examples about why it's also a problem. These are a visit that I did to a TB hospital, rural TB hospital in Malawi. All are TB patients. And I think that the worst place that I visited in my life was this hospital. This is a 3,500 bed TB hospital, and they do a 2,220 smears every single day, and they do around 50 bronchoalveolar lavages for patients suspected to have drug-resistant TB. So you can imagine the situation. And again, around the globe, it's not only affecting specific countries around the globe. You can see here images of rural areas. They don't have electricity, they don't have running water, and they're the primary attention of drug resist for tuberculosis diagnosis, and that's the reason why tuberculosis is still an issue to implement molecular or even phenotypical diagnostic tests in these places. And here an example about US, okay? US that is one of the probably richest countries in the globe. I was looking at in the news different recent cases of tuberculosis be diagnosed around US, and you can see that there are many, including drug-resistant TB, being diagnosed in the US. So it's everywhere. So why is TB a problem? Because it's very easy to transmit, as all of you know. So exposure to tuberculosis just happen if you are exposed to a person that has a cough. It has been shown that in tuberculosis can be in suspension in droplets for 48 hours in a room, if this room is not very well ventilated. So this increases the risk of transmission. And you know, as MTB reaches our lungs, it's in contact with three different environments. One is the lung mucosa, one is the host cell, and then the formation of the granuloma to contain the disease within the lung. So one thing that we're studying in the lab is the first point of interaction, that is the alveolar space. As tuberculosis reaches the lungs, it's in contact with the alveolar space. That is where the exchange of air happens. So the alveolar space, as I mentioned, main function is air exchange. So there are very little cells. It's thought that they have around 12 alveolar macrophages, and it's constituted by around 40 alveolar epithelial cells type 1 and around 67 alveolar epithelial cells type 2. And the volume of lung mucosa within each alveolar sac is around 15 microliters. Now, if we look at the composition of the lung mucosa, that will be the first environment that in tuberculosis it's embedded before it encounters the host cell. The composition is mainly lipids, many phospholipids, but also there is around 5 to 10% proteins, mainly surfactant proteins and hydrolytic enzymes. Now, these hydrolytic enzymes, their main function is to help to recycle the lung mucosa every 18 hours within our lungs. So they are degradative enzymes. We found that these enzymes, when you look at the constitution of these enzymes, they have the activities that are capable of modifying the cell wall of Mycobacterium tuberculosis and any microbe as we know it. Now we have been heavily working in the lab defining these alveolar hydrolyses and their consequences, and also we are focusing in other components present in the lung mucosa in the lung that have been shown to interact with Mycobacterium tuberculosis and how this affects how tuberculosis is infecting the cell. So our hypothesis is that as tuberculosis gets embedded into the lung mucosa, there are these hydrolytic enzymes, here represented by scissors, that modify the cell wall of tuberculosis as we know it, allowing other components of the lung mucosa, like surfactant proteins or components of the complement, to bind differently, as we were expecting, to the cell wall of MTB, and then MTB will interact differently with the cell. So questions that I'm going to try to answer today and convince you is if these hydrolyses can modify the cell wall of MTB, if MTB metabolically adapts to these modifications, and what are the consequences of these modifications, interactions of MTB with host cells in vitro and in vivo. So the first thing that we did is take Mycobacterium tuberculosis and we double radiolabel it. And we expose it to lysates from myeloid cells present in the alveolar space like macrophages or monocytes. And also we expose it to the lung mucosa at the physiological concentrations within the human lung that is thought to be around 1 milligram of phospholipid per ml. We incubated for a time of 12 hours at 37 degrees. Also, we did this up to 15 minutes and we also see the same differences. And after being exposed, we gently centrifuge to remove the bacterium, and what has been released from the bacterium, we add chloroform to obtain two different phases, an hydrophilic phase, aqueous phase, or a chloroform phase hydrophobic fragments, where will be the fragments that are mainly lipids of the cell wall, and we run a thin layer chromatography. And here I'm showing you the autoradiogram of this thin layer chromatography study, in which you only will see fragments, radioactive fragments that have been released from the cell wall of bacterial tuberculosis upon contact with the different samples. So in number 1 and number 5 here, we're showing when MTB is exposed to saline. And we put this as a control because saline is the buffer that we use to perform the bronchoalveolar lavage and to obtain the alveolar lining fluid. Here, I will call it from now on ILF. And you can see that there is, just by the pooling effect of the salt, there is some material being released from the cell wall of bacterial tuberculosis. Then we expose it to monocyte lysates as our negative control because monocytes are thought to have very low hydrolytic enzymes within them. And you can see that similar pattern of what is being released just by contact with salt. And then as for positive control, we expose MTB to alveolar macrophage lysates, very heavily rich in hydrolytic enzymes within them. And you can see a lot of material being released by this contact. And then to alveolar lining fluid, and our surprise was that we saw a lot of material being released just by putting in contact with the lung mucosa of humans, MTB, and this material that is being released was different to the material being released when it's exposed to the enzymes present in alveolar macrophages. What was quite interesting to us. To verify our findings, we took MTB after being exposed to the lung mucosa, and we did sugar analysis in the bacterium itself after being exposed to see if there were changes in the cell wall of Mycobacterium tuberculosis. And we focus in different sugars, arabinose, mannose, myo-inositol, and glucose. All these sugars are main components of virulent factors defined in the cell wall of Mycobacterium tuberculosis like liporabinomannane or trialosimicolate. And we saw that after exposure to lung mucosa from two different humans, healthy humans, we saw that there was a decrease in arabinose and in mannose, in myo-inositol, and also in glucose. Here, as a control, we have a mixture of the enzymes present in the lung mucosa, and also we saw the same outcome. So indicating us that there is some modification in the so-called Mycobacterium tuberculosis. To further confirm this, we did flow cytometry to look at exposure of LAM or trialosimicolate on the surface of Mycobacterium tuberculosis after being exposed to the lung mucosa, and we saw that there was a decrease, around 60% decrease of the presence of MAL-LAM, or around 40% decrease of the presence of trialosimicolate on the surface of MTB after being exposed to the lung mucosa, indicating again that the cell wall is being modified. So this is how we define the Mycobacterium cell wall as MTB is growing in a plate. Has been used in many textbooks. We have the plasma membrane, the peptidoglycan covalent linked to the rabinogalactan, covalent linked to the glycolic acids that are perpendicular to the plasma membrane. And then we have on the top all these virulent factors that interact with the receptors of the host cells. And this is how we define the cell wall of MTB after being exposed to the lung mucosa. And we observe this up to down to 15 minutes if somebody thinks that 12 hours is too long to be not being captured by the host cell. So by 15 minutes exposure, we see that there was a significant decrease on the surface of man-lung, triglyceride, triglycerides, free fatty acids, and many other components, phospholipid 1. All these are being significantly decreased in the cell wall of Mycobacterium tuberculosis. So which implications this has? How MTB is adapting to the lung mucosa after this exposure. So what we did here is transcriptomics, in which we took M. tuberculosis Hermann, that is a laboratory strain, a virulent laboratory strain, and we exposed it to adult ILF or adult lung mucosa from 3 different humans, or to ILF from 3 different elderly humans. Okay, the adult humans defined from 18 to 45 years old, and elderly humans when they are 65 and older. And we look at genes implicated in the biosynthesis of the cell wall after the exposure of 15 minutes. MTB is reacting, and you can see that a lot of genes that are related to the biosynthesis of the cell wall are upregulated when MTB is exposed to the lung mucosa from adult individuals, and actually is not reacting and is not changing too much in the expression of the genes when it's exposed to the elderly ILF, and we're going to visit this later. And just to give you some example, it's not only these specific biosynthetic genes. You can see that MTB is not reacting at all to the exposure to the lung mucosa from elderly individuals, and is reacting to when it's exposed to the lung mucosa from adult individuals. And these drive us to the consequence that maybe the elderly ILF may be dysfunctional of this, or we have some dysfunctionalities that allows MTB not react to it. And it's one thing that we are going to address. So right now in the lab, we are trying to define the interactome, defined as which components of the lung mucosa interact with tuberculosis, changes its cell wall, changes its metabolism, and changes how MTB interacts with the host cells, allowing different outcomes. So we're trying to try to understand why a person may drive primary infection or may drive latency, for example. So what implications do these cell wall changes have in interaction with MTB with host cells? So in studies that I'm not going to show you today, but I'm going to summarize here what we have been doing over the years, when we take MTB and we expose it to adult lung mucosa and we do interaction with macrophages and neutrophils, we see that in both cases, both of them differentially recognize MTB. In the case of macrophages, they recognize less MTB. In the case of neutrophils, they recognize better MTB. However, in both cases, both macrophages and neutrophils trigger a pro-inflammatory response, and both of them can control MTB better by intracellular means. Both of them have less an oxidative response being produced, and they control the growth because there is an increase of intracellular PL fusion. So there's a different trafficking allowing the phagosomes that contain M. tuberculosis to fuse with lysosomes and control better. This is allowing macrophages and neutrophils to control better the infection intracellularly. And also we observe that MTB after being exposed to ILF reduces the extracellular killing mechanisms that the neutrophils have. So allowing and reducing inflammation. And this makes sense to us because the lung mucosa is there to help us, to help us to battle microbes. So what is happening here is that it's increasing the exposure of MTB to the lung mucosa is increasing the mechanisms that cells have to control intracellularly the infection and reducing the harm of extracellular killing mechanisms that neutrophils, for example, have that also will cause tissue destruction. Apart of that, there are also other type of cells that tuberculosis is interacting with, that they are the non-phagocytic cells or very epithelial cells. And we're also working in the lab now to define if exposure to the lung mucosa, of NTB to the lung mucosa, is driving a pro-inflammatory outcome or an anti-inflammatory outcome, allowing these alveolar epithelial cells to control better the infection. And this is ongoing work in the lab. But let me revisit, as I mentioned before, the comorbidity of aging. Why, as we age, we are susceptible to respiratory infections, and in this case, Mycobacterium tuberculosis infection. And we already have postdocs and techs in my lab working on this project. So from now on, I'm going to define the parameters I'm going to present as follows. The lung mucosa, as I call it, will be alveolar lung fluid. It's being collected from healthy individuals that they are, we define it as adult, that are 18 to 45 years old, and elderly will be 65 years old or older. Each M value will represent a human primary macrophage from different donors when we do infections in vitro, and also will represent a different human ILF being used. And macrophages that are used in this study I'm going to present you, they are both. They are monocyte-derived macrophages, obtaining monocytes from blood and deriving between two macrophages, or will be alveolar macrophages directly obtained from the lungs of our donors. And our donors will be adult individuals, healthy adult individuals. For the in vivo experiments, the M value represents different ILFs and also different infection, in which infection we have 5 mice per group and time point. And the mice being used are young mice, 3-year-old mon, and they're the wild type normally used for the TB experiments in the lab. So the first thing that we did is to study the composition, the cytokines, and the complement, and the surfactant protein levels. So all components that have been shown to be impacting the interaction of MTB with the host, and we study the adult and the elderly lung mucosa. And when we normalize by the same protein content, we're observing that the elderly lung mucosa has a pro-inflammatory signature, as marked here by the IL-1 beta related to the inflammasome, or the Th2 cytokines, pro-inflammatory cytokine IL-6, or Th1 cytokine TNF-alpha. We also observe that there is an increase in some of the components of the complement in the lungs of the elderly individuals, especially the alternative pathway of the complement. We observe also that surfactant protein A and surfactant protein D, that are critical innate modulators to allow the host cells to control better the infection, they are also increased in ILF, which puzzle us because we know that elderly individuals are more susceptible to TB infection. So we then went further and studied the binding of these components. And we found that even if they have higher levels in the elderly, the binding to Mycobacterium tuberculosis is lower. So this means that these proteins, even if they are elevated in the lung mucosa of elderly individuals, they are dysfunctional. And this may be due to the fact that when we measure the oxidative stressors in the lungs of the adult or elderly individuals, we observe that there is more oxidation in the lungs in the elderly individuals that may influence how these proteins are dysfunctional. So of course, we cannot measure directly ROS, radical oxygen intermediates, or nitrogen intermediates, but we can study their footprint left behind. And in this case, here we measure the carbonylation of proteins or nitrotyrosination of proteins that relate to nitrogen intermediates or radical oxygen intermediates, and you can see that the proteins are more oxidized, and this can drive the dysfunctionality of these critical components in the lung of the elderly individuals. We further performed mass spectrometry analysis looking and try to understand what are the composition of these proteins that they are oxidized within the lungs of elderly individuals. And here we have just a volcano plot showing some of these proteins that are related to tissue destruction in the elderly, that they are way upregulated. And we were looking more in detail the composition of the elderly lung mucosa versus other lung mucosa. We are observing that there is an increase of pro-inflammatory and pro-inflammation proteins in the lungs of elderly individuals, and there is a decrease of anti-inflammatory proteins in the lungs of elderly individuals when we compare it to the adults. And also there are an increase of antiviral peptides, but again, we show that some of these ones are dysfunctional. So when we make the complex analysis of what these proteomics results mean, and we do pathway analysis to try to determine which pathways are implicated in driving susceptibility of the elderly to respiratory infections, focusing in tuberculosis, we are seeing that there is an upregulation of the remodeling of the lung epithelium, and also there is different pathways that are related to try to rebuild the lung mucosa and the cell-on-cell interactions in the lung mucosa of the elderly that may implicate why the elderly are susceptible to respiratory infections. And focusing in these respiratory abnormalities, we identify this network. And just as an example, I'm not going to go through, but you can see that there are specific targets that now we identify that we can target to try to reestablish their function or try to knock down their function, that drives that an elderly have an abnormal situation in the lung that may favor respiratory infections. Keeping this in mind, we took Mycobacterium tuberculosis and we exposed it to adult or elderly ILF, being adult ILF, the white open bars, and the elderly ILF, the black bars. And we infected, as I mentioned before, macrophages, monocytic macrophages, or alveolar macrophages. And we can see that after being exposed to the lung mucosa, tuberculosis can infect and can grow faster within macrophages. You can see here the difference in growth is around 60% different in growth and 86 hours. This is in vitro. What happens in vivo? So when we take tuberculosis and we expose it to the lung mucosa from adult or elderly, and we infect mice, at 2 hours post-infection, when we euthanize the mice and we take their lungs and we look at the CFUs, we can see that there is similar amount of bacteria, but this bacteria at 21 days or 60 days post-infection within the lung, you can see that it grows faster. So in the case of the MTB exposed to the adult ILF, the mice can control better the infection. In the case of being exposed to the elderly lung mucosa, the mice cannot control the infection. And this is like 1 log and a half, one log difference. That is quite substantial for the case of MTB in mice. And the same was observed in the spleen as an indicator of systematic infection. When we look in deep in the lung and we look at inflammation, we can see that when it's exposed, when MTB is exposed to the adult ILF, you can see that there is some inflammation at day 60 and increasing at day 150, but there is more focalized and there is more inflammation when MTB is exposed to the LDL ILF, indicating that MTB is favoring the infection within the lungs of the mice, as you can see it here. And when we look at survival of the mice, the mice succumb to the infection faster when it's exposed to the elderly ILF versus the adult ILF, indicating that some factors in the elderly ILF drives MTB to become more virulent or to grow faster, and drives the inflammation that is the hallmark, and tissue destruction that are the hallmarks of TB disease in the lungs. So as a conclusion of the first part of the talk, we define that there is a basal higher levels of oxidation and inflammation in the lungs in the elderly individuals, that these elderly individuals have critical innate soluble proteins that they are dysfunctional, that have been defined to help to fight tuberculosis infection. So they don't work very well, and we think that is due to the oxidation status of the lung in the elderly, and that when MTB is exposed to the elderly, to the lung mucosa of elderly individuals, favors its growth in vitro, in human macrophages, and in vivo using the mouse model. So next, we thought, well, another comorbidity that is very important is HIV. So how HIV changes the lung environment in the lung? We know that also HIV individuals are susceptible as elderly individuals to respiratory infections and to MTB. So we went over and analyzed the lung mucosa from HIV, from people living with HIV. And here are our cohort of people living with HIV and their characteristics. They were age-matched, the healthy, as we're going to call them, and people living with HIV, donors of the lung mucosa, or donors. They did not receive antiretrovirals. They have a medium count of CD4s, or 124. This is their viral load median. The age median was 36, and they have a median BMI of close to 19, and this is the ratio of male versus female. So when we first analyzed our cohorts, we observed that, again, like in the case of the elderly, the people living with HIV had a very high oxidation in their lungs, here represented by analyzing the proteins that have been carbonylated or they have been nitrotyrosinated. However, we did not see differences in their oxidation status due to the presence of myeloperoxidase. That is one of the major enzymes related to the oxidative stress induced by neutrophils migration into their lungs. When we look at the cytokine profiles, contrary to what we observed in the elderly, remember in the elderly everything was up. Here in the people living with HIV, their pro-inflammatory status in the lung is decreased. We saw a decrease in Th1, Th2, and Th17 responses when compared to healthy, and some of them quite significant, like in the case of, you can see interferon gamma, TNF, IL-2, and IL-10. When we look at the function of the levels and function of some components of the complement or the surfactant proteins, we saw that looking at the levels also they were decreased. So contrary to what we observed in the elderly, that they're increased but they're dysfunctional, here they are decreased, and also some of them are dysfunctional, especially in the case of surfactant protein D. We can see that they have lower levels, and the levels that they have, this protein is dysfunctional in their lungs. So when we did proteomic analysis of the component of the proteins present in the lung mucosa of people living with HIV versus healthy, we saw that these people have an increase of proteins linked to inflammatory pathways. We saw that they have a decrease of antioxidant proteins, and importantly, we saw that they have practically the complement system in their lungs is shut down. Everything was very low versus healthy individuals. And this may be related to a decrease in these two factors here that are thought to regulate the complement system, like PGR and PCJ. Next, we wanted to see if we see any differential association of MTB after being exposed to the lung mucosa of adult individuals that are healthy versus people living with HIV. And in this case, we took M. tuberculosis fluorescent red tomato and we exposed it to healthy ILF from adult individuals versus GFP MTB exposed to HIV lung mucosa from people living with HIV lung mucosa. And we mix 50-50 the bacterium and we infect a monolayer of macrophages. And you can see that after we do a 50-50 infection, the bacterium that has been exposed to the HIV lung mucosa binds better, associates more to macrophages than the bacterium that has been exposed to the lung mucosa from an elderly individual. That is quite interesting because telling us that is more infectious, that NTB becomes more infectious after being exposed to the Lyme mucosa from an HIV individual. When we look at the intracellular growth within these macrophages, we can see that after being exposed to the Lyme mucosa from HIV individuals, NTB grows faster. And this is a representative experiment, and this is an accumulation of different experiments showing that NTB grows faster over time after being exposed to the Lyme mucosa from people living with HIV. When we look at the mechanisms that could drive this increase in growth within the macrophages, and we look at trafficking, we have NTB here being exposed to healthy Lyme mucosa, and you can see here is GFP MTB within a phagosome, and here we stain for lysosomes with the LAM1 marker. And when the phagosome fuses with the lysosomes, it becomes yellow, and you can see that there is a phagosomal fusion that drives the killing of MTB. And in the case of HIV, MTB exposed to the Lyme mucosa from HIV individuals, NTB, you can see that does not fuse with the lysosomes, but means that NTB will be able to replicate or stay alive within the macrophages that they're infecting. And you can see here a significant decrease in the colocalization of NTB within the phagosomes and the lysosomes when NTB is exposed to the Lyme mucosa from HIV infected individuals. So the thing that we did then is how we can restore this, how we can make that the Lyme mucosa from an HIV individual gain functionality and gain the capacity to control tuberculosis infection. And we focus in surfactant protein D because this is one of the innate modulators that we observed in the Lyme mucosa that was heavily decreased. And in this case, again, using different Lyme mucosas and different cells, we observe exactly the same phenotype that before. NTB exposed to the Lyme mucosa from healthy individuals with the dark bars, and in green infected with HIV, you can see that grows faster. When we replenish and we add surfactant protein D to the healthy, because surfactant protein D in the healthy is functional, we did not see any differences in the capacity of macrophages to control NTB that have been exposed to the Lyme mucosa from healthy individuals. However, when we replenish the Lyme mucosa from people living with HIV with SPD, you can see that there is a gain in function and the macrophage can control again the infection better at the same level that a healthy individual will do. Concluding that SPD is a critical factor that we should be studying that allows people with HIV to control better respiratory infection, in this case, Mycobacterium tuberculosis. And this is due, what we found today, because we can reestablish the capacity of the macrophage to control the infection by increasing phagosomal lysosomal fusion. So again, as before, MTB exposed to the Lyme mucosa from a healthy individual, where here fuses and macrophages can control better the infection, here in yellow. From HIV individual does not fuse, so they cannot control better the infection. And when we replenish with surfactant protein D, you can see that the macrophages reestablish their capacity to increase phagosomal lysosomal fusion and control the infection, as is shown here in the graph. So in our conclusion here, we find that we have a healthy individual, one elderly individual, one person living with HIV. We have NTB reaching the lungs, getting embedded into the Lyme mucosa. The Lyme mucosa in a healthy individual that is young, an adult individual, is functional and can, modify the NTB cell wall, allowing other components to opsonize the bacterium differently. This opsonization allows the macrophage to take tuberculosis. There is an increase in phagosomal fusion and allows better the control of the infection. In the case of the elderly people with HIV, if they get infected with MTB, the ILF is dysfunctional, does not work well, does not opsonize these components, do not opsonize very well the bacterium. The interaction with the macrophages is differently, allowing MTB to grow and replicate faster within macrophages, maybe then allowing to establishment of the infection better than in the case of the healthy individuals. So now to finalizing the last 8 minutes that I have is, can we revert this? Can we come up with a concept in which we can take and we can change the oxidative status of the lung of elderly individuals or people living with HIV, and by a treatment allow them to restore their capacity to control respiratory infections? And this is what we're working now in the lab, and I'm going to show you some of the current data that we have. So in this case, this is established in the field that as we age, during the continuum of aging, arrive to one moment in which inflammation in the periphery or inflammation in the lungs increases. It's thought that inflammation in the periphery happens before, as represented here with the open circles, and in the lung is a little bit late. But arrive to one moment, what we call it the tipping point, maybe between 50 to 60 years old, in which the inflammation starts to increase. So we want to see if an intervention prior, around 30, between 30 and 40 years they are old, will allow this to be flattened and doesn't increase, or being delayed so we can reach the elderly age being much healthier. To do that, we use a system that we use a technology called nanotechnology, in which we microencapsulate different components. We use these nanoparticles because they are very stable. And we took the nanoparticles and we encapsulated rapamycin, that is a modulator of oxidation in the lungs, and is now defined to reduce cellular oxidative stress and remove damaged cells from the lungs. Also, we add a mixture of antioxidants that I cannot describe here because it's under provisional patent, and now my collaborators will kill me. And also we added surfactant protein D as one of the major proteins that we want to establish in the lungs. What we did is encapsulate these components with nanoparticles, and we did this because these nanoparticles allow us to have a slow release of these components in the lower space. As we're seeing here, when we take antioxidants or rapamycin and we deliver it into the lungs of mice, and then we perform bronchoalveolar lavages and do the pharmacokinetics, looking at the concentration of these antioxidants and rapamycin within the lungs of the mice that receive these nanoparticles intratracheally, we can see that the concentrations of these two components in different experiments are quite stable for around 14 days at a concentration of 3 micrograms per ml. So we went with this approach. And then we tried different approaches, administering to the mice twice a week the nanoparticles, or once a week, or once or twice a month, only once a month. Surprisingly, we found that giving it once a month and then analyze after 1 month what happens in the lung of these mice was the best approach to do. So as a result, when we treat mice with these nanoparticles, and here we have vehicle, that are the empty nanoparticles, young mice, vehicle, treated all mice, and the all mice treated with the combination of antioxidants, rapamycin, and surfactant protein D nanoparticles, looking at the oxidation status of their lungs after 1 month of the one single treatment, we can see that there is a reduction of the oxidative status in the lung of the all mice that receive the vehicle, empty nanoparticles, versus the ones that receive the loaded nanoparticles. But we look specifically at oxidation, looking at oxidation of the mitochondria within the cells in granulocytes, lymphocytes, or macrophages. We can see that there is a decrease in the oxidative status of these cells in the all mice that receive this treatment. When we look at cytokines that are linked to inflammation or to proteins that are linked to tissue destruction within the lung, as we age, we can see that there is a reduction of inflammation and there is a reduction of tissue destruction in the lungs of elderly mouse that have been treated with these nanoparticles. And when we then took these treated mice and we infect them with Mycobacterium tuberculosis, you can see here at 10 days post-infection, some mice already can control better the infection, and this become significant at 40 days post-infection. And we observe this is only repeated once, so we need to repeat it more times in this case. But you can see that we can see that the mice can control the infection with close to a half-log reduction in the lungs. That is the same protection that the BCG vaccine is providing in the mouse model. When a mouse is vaccinated with BCG and then infected with TB, also we observe this half-log protection. So here, just by treating the mice with the nanoparticles, we can see that they can be better protected against tuberculosis infection. So as a conclusion on my last slide, we found that, that in the elderly environment, there is a high oxidative stress in the lungs that drives innate dysfunction. And data that did not show this is because there is a downregulation of specific regulators of inflammation that drives this innate dysfunction, and this oxidation go very high as we age. That drives an increase of the pro-inflammatory and inflammation in the lungs as we age. And then that by treating with nanoparticles, we're able to control this, and maybe would be a remedy to improve respiratory resistance to respiratory infections. In the case of HIV, our current hypothesis and the results show the same, that there is a high oxidation and a high immunodysfunction. However, the expression of negative regulators is high in comparison with the aging, and this drives that there is less inflammation and less inflammatory responses in the lungs of HIV individuals. And again, we are now in the process, and data did not show, of to see if using the humanized mouse model we can control, and doing the nanoparticle treatment, we can control, also allow mice infected with HIV control better the infection. Finally, I just want to thank the members of my lab and our collaborators and pulmonologists that have been implicated to provide us the lung mucosa, and also our funding sources, and I will be very happy to answer any questions that you may have. Thank you.
Muchas gracias, doctor. ¿Tenemos alguna pregunta de la audiencia?
Thank you very much, Doctor. Do we have any questions from the audience? The first question would be, with regard to these changes in the expression of a comorbidities, of a clinical relationships, have you studied that?
Al menos hemos mirado.
We have seen HIV, we have seen in the elderly, and we are working with Dr. Blanca, we're working on diabetes, and we are seeing a relationship between dysfunctionality and these immunomodulators present in the lung mucosa with the resistance of people with diabetes or HIV. With regard to the use of drugs, are there any changes from these drugs? We haven't seen that, but that's a good question. What we do know is that there are components, there are specific antioxidants that I can't say because they're under patent, but they reduce oxidation of the lungs. We have seen that in people with in smokers and ex-smokers who have been treated, and we've seen a reduction of lung oxidation in these people, and they improve their breathing quality, their lung function. And now we are evaluating if they would be more resistant or not to infectious diseases, to see if they establish their innate lung function. It's interesting. Is there a correlation between these changes in the bacteria with regard to the different lineages of tuberculosis? Yes, we have seen, in what we presented today, we've seen different lineages. For example, Beijing lineage with HN, HA, and we've seen different multi-resistant strains which react differently. So depending on the lineage and regardless of the profile, they react differently when they are exposed to a lung mucosa. We've seen in TB that they react very heavily. Reacciona.
Enzymes present in the lung mucosa alter the cell wall, and because they have a thicker cell wall, they react heavily. In the case of hypervirulent strains of NTB like HN878, that is a Beijing lineage, do not react. Doesn't matter which lung mucosa you are, adult, elderly, diabetes, HIV, do not react at all. They react similarly to bacteria grown in plate. That is quite interesting.
¿Tienen plan de implementar el análisis entre...
You have plans to implement analysis between different different populations like Latinos? I would love to. We are in conversation with Dr. Adrián Rendón and Raquel to see in South Africa. And in South Africa, the problem we have in South Africa is, in addition to the fact that they are exposed to a lot of smoke and they have a lot of dysfunction in their lungs. So all the studies that we have is they are very low. And what we have in tuberculosis and in the lung function of people in South Africa, and they grow.
Hipervirulento dentro de los ratones.
And they become virulent, and we suppose that is because even constantly they are deregulating some of the functions which are necessary in the lungs of people to better control infections. With respect to the other extreme and the children, with children we did a study of COPD and EM.
And we expose it to a mucosa from pediatric children that have COPD. It grows faster and establishes biofilms faster than when it's exposed to Lyme mucosa from healthy children. That also blew off our minds, indicating again that this dysfunctionality of some components in the Lyme mucosa due to COPD that these kids have drives this, and also in no COPD, sorry, cystic fibrosis, with fibrosis cística, con niños con fibrosis cística, sorry.
Children with cystic fibrosis, sorry. Do you have a characterization of the microbiome of the lungs in this case?
Tenemos las muestras y estamos...
No, but this is a good question. We have samples and we are talking to lung experts who are interested in studying the microbiome of the lung and the interrelationships between with what we are observing to see if by manipulating the microbiome in the lung, we have both of them, bacteria and the viruses that there are in the lungs, how we can manipulate these to see if we can, again, if we can find out the innate function that is critical for controlling infection in the lung. Do you have a characterization of the growth of pathogenics? No, pathogens? No, we haven't studied that. We have colleagues who are in that environment, but if anybody wants to do that and you need samples, we would be happy to work together with you. Thank you. That's the idea of this forum, to foster these collaborations. One last question is, how is the alveolar liquid collected? And how do you have the replacement of the SPD in the laboratories?
What we do is the following. So we perform bronchoalveolar lavages. All pulmonologists do 3 washes. We get around 80 mLs, around between 60 to 80 mLs of bronchoalveolar lavage, and this is extremely diluted, does not mimic what is within our lungs. So then there are studies show that the concentration of the physiological concentration of the lung mucosa within the lung has 1 to 1.5 milligrams of phospholipid per ml. So we take the bronchoalveolar lavage and we concentrate it to this concentration of phospholipid. And then we take mainly this 80 ml gets reduced between 20 to 34 reduction. And we finish with 1 to 2 mLs to mimic the concentration within our lungs. And then we take MTV and we expose it to this concentrated lung mucosa that we call it alveolar lung fluid.
Okay.
And respect the clinical studies, we don't yet. We are working with some pulmonologists to obtain funding to do these studies. I think that the in vitro studies and the in vivo studies using mice shows it. Right now we have studies using the non-human primate model of a simian HIV infection, the simian immunodeficiency virus infection, and also we use the baboon model for the elderly. We are seeing similar patterns like humans, elderly non-human primates present dysfunction of these components in their lungs, and now we are start to do the experiments in non-human primates that will allow us to obtain the necessary preliminary data to even go to the FDA to do some clinical study in humans. If not, without the human primate studies in the US, we cannot move forward. That's a great question. We will love it. We love it to do.
La siguiente pregunta es con respecto a estas nanopartículas.
The next question is about these particles. They are attacking these locally. Is there some kind of systematic approach? Yes. We were, as we were saying in the lung, we used to think of, we could attack the lung systematically. But we have a systematic intervention, which would be easier. And then we could see how this systemic intervention affects the lung. What we thought was we are developing a kind of spray which people could apply directly in their mouths to so that it could reach the lung. These components would need to arrive to reach the deepest parts of the lung, and they would dissolve bit by bit into the lung mucosa over time. But systematically, that would be a good concept to implement that more easily. Thank you. Thank you very much. Very interesting and novel. We have one question from Dr. Laniado. Yes, go ahead. Can you give us your comment about the plasma levels? These tests, I usually think of them when we have a patient that is not responding to treatment based on a scheme that was based on sensitivity tests. We did schemes, but the patient was not responding based on the sensitivity tests. The first thing that we would ask is if the patient is taking the drug, and we cannot guarantee that. So that's the first thing that we would look at, whether the patient is really taking supervised treatment. In Mexico, we use flexitized or semi-supervised treatment, where the patient is given once, we see the patient once a week, and we think the patient is going to take the drug. So there would be a resistance. Initially, we had a pattern, but this was changed, and so that's why the patient is not responding. The third possibility would be that we have lesions where the drug is not reaching. We have patients where the resistance pattern is not changing, but we have a lesion. We did a mammograph, for example, and we see that the blood is not reaching that area of the lungs of the drug is not reaching. So that would not be able to eradicate the bacillum. And then lastly, we have absorption. That's not very frequent.
El problema que tenemos es que no es fácil hacer las pruebas, no cualquier laboratorio las puede hacer. Nosotros en algún tiempo estuvimos haciéndolas a través de Estados Unidos en el laboratorio del Hospital Judío de Denver y son bien costosas. Entonces sí tienen su lugar. De hecho, Raquel y tú estabas en este equipo, hicieron medición de rifampicina, por ejemplo, de niveles séricos, y nos dimos cuenta que pues todos los pacientes, no solo los diabéticos, que era lo clásico de la literatura, tenían niveles subóptimos de rifampicina. Esa puede ser una razón también. El paciente pudiera estar recibiendo, no porque no se tome la medicina o porque no la absorba, sino que de hecho la presentación de dosis fija combinada nos hemos dado cuenta que generan niveles subóptimos de rifampicina en sangre. Entonces sí se pueden utilizar, pero primero habría que descartar las otras posibilidades que son más frecuentes, ¿no?
Muchas gracias. Muchas gracias a los dos ponentes el día de hoy. Vamos a concluir ahorita ya la sesión de preguntas y agradecemos nuevamente al Centro de Estudios y Producción Audiovisual CEPA por el apoyo en la transmisión de este evento y también a los intérpretes del idioma inglés. Le recordamos a todos los asistentes registrar su asistencia en el código QR que estuvo apareciendo durante la sesión con el fin de contar su asistencia para la constancia. Solo es válido hasta el final de esta sesión. Agradecemos la asistencia a esta sexta sesión y los invitamos a la siguiente semana para la séptima emisión de este Foro de TV con dos excelentes ponentes, el doctor Feliciano Milán Suazo, con la plática Vacunación contra la tuberculosis bovina, experiencias de investigación y perspectiva de uso, y el doctor Francisco Olea Okelka, con la plática Using the One Health Approach to Others to Changes Posed by Zoonotic Tuberculosis. Muchas gracias a todos.
Gracias.